Afternoon, and thank you for coming to my talk, Leveraging Deep Learning for Characterization of Malaria Parasite Puffs. We've come up with this acronym for proteins of unknown function, and we call them puffs. So in 2022, there were an estimated 249 million cases of malaria worldwide with 608,000 deaths. And on the left, you can see the plot for incidence. And incidence here is a metric that defines the number of new malaria cases in a year per 1,000 population at risk. From this plot, we can see a density in sub-Saharan Africa, but also prevalence in large parts of Asia and in South America. Malaria is caused by this parasite, Plasmodium falciparum, and that'll be abbreviated as PF for the rest of this talk. And in 2002, its genome was sequenced for development of diagnostic markers and therapeutics. But when we look at the genome and we look at the scope of Go annotation that's found within the proteome, out of the 5,495 protein coding genes, we see that 1,270 of them are unannotated across all Go namespaces. So molecular function, biological process, and cellular component. And these are the proteins that we call puffs, so proteins of unknown function. There's also 41% of these proteins that are partially annotated. So they have an annotation in one namespace, but they might not have an annotation in another. So at the core of trying to find new diagnostic targets and therapeutic interventions for malaria, we really want to look at these puffs and try to see if we can come up with annotations that can help malaria biologists in the lab. And so a simple metric that we did first was we decided to use sequence similarity. And in this process, we took our puffs, the Plasmodium falciparum puffs, and we blasted them against SwissProt. And what we found was that for 90.2% of these proteins, there were no hits. And for the 9.8% of proteins that did have hits, there weren't enough confidence in that in order for us to allow to transfer the annotation. And now this, along with other data that we have, can help us make the claim that sequence similarity is not sufficient for annotation transfer, at least when it comes to these falciparum puffs. But if we zoom out for a second and we look at the sequence structure gene ontology relationship, we have data that indicates that the PF puffs lack annotated sequence homologs, which makes it difficult to exploit the sequence to gene ontology relationship. So our work in our model, Plasmodium function predictor, works on the structure to gene ontology relationship. A core question, though, that we have is that how can we represent structure in an informative way in the context of a model that allows us to transfer annotations over? And the way that we do that is by using a model that our lab published last year called TMVec. And TMVec takes and relies on fine-tuning a pre-trained language model representation for a structural similarity learning task. Now, what this briefly means is, given sequence X and sequence Y, the embeddings that are produced by the pre-trained language model are processed through a twin neural network called Phi. And Phi produces vector representations of these initial embeddings, where sequences that are structurally similar are closer together in the latent space, and sequences that are structurally dissimilar are further apart. And we can visualize these vector representations to get an idea if these new representations are actually encapsulating structural information. So here we have on the left the default ProTrans embedding, and in the left panel, and on the right panel we have the TMVec embeddings. So when we look at structural categories that are present on Cath, we see that TMVec is robustly separating out some of these hard to differentiate structural categories. And this gives us an idea, along with the rest of the paper, if you'd like, you can read it, that these embeddings are encapsulating the structural information for these particular sequences. And so that's what we use as the bedrock for our model. But given that we had a difficult time trying to annotate using sequence to gene ontology, given that Plasmodium is so diverged from the rest of eukaryotes, we also wanted to focus on relevant structure-function relationships. So here we have Plasmodium lies in the eukaryotic supergroup called SAR, and this is highly diverged from where most function prediction models are trained, can be found down there in the green. In the context of function prediction, when we use these general function prediction models, we find that they have a difficult time providing specificity in Go terms when we're feeding them our SAR sequences. And that just, I think, is a function of the fact that these sequences are so diverged. So our model is called Plasmodium function predictor. And Plasmodium function predictor is a collection of 3 models that are trained on the 3 separate sub-ontologies: molecular function, biological process, and cellular component. We have PFP, MF, BP, and CC. We can dive into the architecture for what this model looks like, and also but it's the same for across all of our sub-ontologies. So here we have the architecture for PFP-MF. And given an input protein sequence, it is processed through our frozen TMVec model layer. And what this does is that this produces an embedding that captures structural properties that are associated with that protein. And this is then attached to a fully connected network, a fully connected layer, which is just a feed-forward network that outputs the Go terms at the end. Given that we're working with a diverged species, we also wanted to see if it was possible for us to implement uncertainty in our predictions as well, because that would be informative for biologists in the lab. And the way that we do that is by leaving this dropout layer on during inference time. So there's two parameters that you can see on the screen. There's gamma and beta. And gamma is the proportion of neurons that are turned off during the in the dropout layer at random. And beta is the number of passes that each sequence will make through. Beta is the number of passes that each sequence will make through the model in order to not only just have a singular softmax probability that's associated with a Go term, but a distribution of softmax probabilities from which we can model uncertainty. Aside from our test set, we also wanted to have a more relevant evaluation, given that we're trying to predict function in Plasmodium falciparum. And so we have this Plasmodium holdout data set that contains manually curated annotations that are found within the Plasmodium genus that are hidden from training. And when we also do a 6-mer amino acid comparison to the training data sets, we see that this data set also differs. So this serves as a twofold challenge for us because we're asking PFP to predict manually curated annotations, but we're also asking PFP to predict sequences, functions for sequences that are dissimilar from what it's seen during training. To evaluate the architecture of PFP and whether the TMVec embeddings that we have enhance the function prediction process, we also trained 2 additional models. So here on the screen, you have SAR-CNN-MF, which is trained on sequence function relationships, and it uses the same training data that PFP uses. So it's trained on relevant sequence function relationships. We next have Go-TMVec, which is a general function prediction model. And this is trained on structure function relationships that uses the TMVec embeddings. And it uses the Cath5 training data set, so it's species agnostic. On this plot, we also have DeepGo Plus, and we've subsetted it just for the molecular function ontology so that we can show you some of the claims that we make. We can see that PFP-MF outperforms all of these baseline models, but we claim these 3 trends. So 1, we see that in the context of function prediction, in the SAR clade, specifying relevant sequence structure function relationships enhances performance, as seen by the improvement between SAR-CNN-MF and DeepGo Plus MF. Secondly, we see that focusing on the structure function relationships instead of sequence function relationships, given the TMVec embeddings, which are encapsulating the structural information, provides an improvement in performance between Go-TMVec and SAR-CNN-MF and DeepGo Plus MF. And lastly, when we combine both of these efforts, so working on structure function and also training on relevant structure function relationships, we see the best performance, which is with PFP-MF. So these plots were just for the molecular function, but this is a trend that we see across biological process and cellular component as well. In terms of metrics for those models, here we compare them to DeepGo Plus. And so we have PFP-MF to DeepGo Plus MF, PFP-BP to DeepGo Plus BP, and so on. And we see that when comparing all of our PFP models against DeepGo Plus, we outperform them when it comes to Fmax, AUPR micro, and AUPR macro. Our MF, BP, and CC models obtain an Fmax between 0.78 and 0.88. But notably, our models still have high unweighted AUPR macro performance, which is a good sign for us. When looking at the second metric in regards to function prediction, SMIN, we see that PFP-MF and BP outperform their DeepGo Plus counterparts, while PFP-CC has comparable performance. But when we dive further into the SMIN metric, and you have remaining uncertainty and misinformation, we see that PFP models have lower misinformation than their DeepGo Plus counterparts, particularly BP and CC. So now that we've kind of established that PFP is good at predicting Plasmodium functions, we can see in how the model is predicting these functions. So given, this is for a given sample sequence within the Plasmodium holdout data set. And since we leave the dropout layer on during inference time, we generate a distribution of softmax probabilities that are associated with each of these Go terms. And that's what you can see on the x-axis. So we pass each sequence through 100 times, and that's what gets us our distribution. On the y-axis, you can see the binarized predictions for this particular sample, with the true label being protein phosphatase binding. So what we see is that these Go terms that we're predicting are following the hierarchical relationships that we see within the Go MF tree. But now if we want to report uncertainty in a meaningful way for our model, we can zoom into each of these softmax distributions and see that they're all skewed right. So what we do is, in order to binarize our predictions, we use the median of this distribution, and then we look at the median absolute deviation as the measure of uncertainty that we have in predicting this particular Go class. So these last two slides were just for a sample sequence within our Plasmodium holdout data set. But what we can also do is that, given a particular data split, we can see which Go terms the model is most uncertain about. And what we find here is that we're looking at molecular function terms, which the PFPMF model is most uncertain about across our test split. We see an overrepresentation of terms here that are associated with binding. And this was an expected finding for us, since PFP uses flattened representations of protein sequences and doesn't have per residue context, which is what some of these terms would be involved in. So they would only be responsible for a couple of residues within a protein in order to get that annotation. But what we see, though, is that all of these terms have low information content, which was a good sign for us. And to see if PFP can not only predict function for our puffs, or our proteins of unknown function, we also wanted to see if it was possible to use PFP to predict for the partially annotated proteins as well. And the way that we do that is that we've come up with this metric, with this evaluation. And here we have a partially annotated protein X that's currently found within the proteome. It has a biological process and it has a cellular component, but it lacks a molecular function. So we can predict the molecular function using our PFPMF model. And then what we do is that we create pairs of these annotations. If we were to predict a molecular function term, given that there's a biological process term and a cellular component term, we'd want it to make biological sense. And the readout that we have for this is to just see if there's other proteins on UniProt that have this pair of annotations. When we run this across the entire proteome, we generate 534 pairs of PMF and EBP and 739 pairs of PMF and ECC. So when we query UniProt to see if these are pairs that we find on UniProt, or proteins that have been annotated with this molecular function and this biological process term, and this molecular function and this cellular component term, we find that 96% of these pairs are found on UniProt. And for the 3-ish to 4% of proteins for the 3 to 4% of proteins where the pairs aren't found on UniProt, they're just Plasmodium specific Go terms, so we wouldn't really expect to find other examples of that. But it allows us to make the claim that PFP can impute biologically plausible annotations for sequences that already have an annotation. So we can predict annotations for our puffs, but we can also help fill in the gaps within the proteome. Our predictions are at least 4 nodes away from the root, which was a good sign for us that we're predicting specific annotations that can be seen across all 3 of these namespaces and the darker sections of these pie charts. But kind of going back to uncertainty, we can also quantify that for a given protein and the set of Go terms that we predict. Are we highly confident? Are we moderately confident? Is it exploratory confidence, or is it an assorted set of confidence across all of these Go terms? To see if any of these annotations have actually been experimentally validated, we used this paper that came out earlier this year that did a screen to identify RNA binding proteins within Plasmodium falciparum. So here what we have on the left is we have specific high and moderate confidence PFPMF predictions for proteins that are lacking a molecular function term. And what we find is that for proteins that we're predicting with nucleic acid binding and RNA binding, these are all proteins that were found within this experimental screen. So it gives us an idea that the predictions that we're providing have they have some, wet lab background. But broadly, what we're seeing here is that PFP models can improve the annotation of the falciparum proteome. This is the same pie chart that you've seen. But when we run PFP on all of the missing partially annotated proteins, what we find is that we can take our puffs from 23% down to about 3.9%, and we can improve the annotations for 35%, for fully annotated proteins from 35% to 61%. And this is after we filter the predictions for proteins that have annotations that are greater than 4 nodes away from the root. So in summary, we have, there's two sections. There's PFP training and architecture. We report the first curated Go function prediction model that is developed for a subset of eukaryotic species and not trained on all general species. And we report the first model to estimate uncertainty with Monte Carlo dropout for our Go terms. And we find that training on relevant structure-function relationships improves model performance compared to baseline models. Now, this is like an expected thing, but we're happy to provide some numbers to this. And in terms of our predictions, we can impute Go terms for partially annotated proteins, and we can predict novel Go terms for PF puffs that are in a biologically relevant context. Our predictions are specific and can be grouped by model confidence, so there's uncertainty. And certain predictions that we have been experimentally verified. But we reduced the Plasmodium falciparum puffs by 19%, and we increased annotated PF proteins by 26%. So going back to the central question at hand, our predicted Go terms can improve the annotation, and they can potentially help aid in malaria diagnostic and therapeutic discovery. And with that, I'd like to provide thanks to my collaborators at NYU, but also both of my PIs, Jane and Rich. And with that, I can take any questions.
Okay, Ido?
Thank you. Great talk. So I have a couple of questions. One is, how much were your predictions informed by the fact that Plasmodium falciparum is a parasite, and in terms of biological process, it has a lot less than most eukaryotes, being a parasite, so you can definitely rule out functions simply by knowing they don't exist in Plasmodium. Is that something you put into your data set?
I guess implicitly, because we focused on just the SAR sequences, that we were focusing on biological processes that would be, found within other parasites as well. So we were taking that into account. Does that answer your question?
Yeah, but do you think that if you did more explicitly, you'll have better predictions?
Absolutely. I think that it's just a lack of training data. I think that when you're working with parasites that are, this diverged, when we had to go up to the eukaryotic supergroup level in order to get enough training data that allowed us to.
Which brings me to the second question about the training data. Plasmodium only has about 5,000 genes, I think.
Yeah.
So how did you expand that?
So the Plasmodium genus is completely hidden from the model during training.
Okay, so the entire Plasmodium.
Yeah, the entire Plasmodium genus is. So it's every other species within the SAR.
Okay.
Within the SAR supergroup.
So how did you prevent within that group? So how did you work on preventing data leakage? Like, did you some % similarity cutoff? Or?
Yeah, we did a sequence similarity split for our test, train, and validation.
Okay.
And we also used the Plasmodium holdout sequences to test our annotation.
Okay. Okay. That's something I didn't get. Thanks.
Yeah.
All right.
Okay. Any other question? No? Yes.
This is a naive question. So I understand that you got better data because you focused on training on the Plasmodium species. But the initial annotation of these, they had to be transferred by a way that would have been crappy anyway because the knowledge was not known. So how do you make sure that your initial annotation, so how did you get better when your training set was based on things that would have to be crappy because they're not well transferred?
Yeah, that's a great question. So the way that we kind of take that into account is two ways. We add manually curated annotations and automatic assertion annotations within our training set, but we weight the manually curated annotations much, much higher than our in terms of the loss function. And so we're able to predict, when we predict, we kind of take that into account implicitly because they matter more. And hopefully our Plasmodium holdout data set also serves as a good proxy because those are manually curated annotations.
I have a question regarding your proteins. Anything about the composition? They have weird compositions, or I could call complexity, things like this. You have those been taken account into your?
Yeah, so it isn't in this slideshow, but Plasmodium is known to have low sequence complexity and a lot of intrinsic regions of disorder, and it's one of the best known eukaryotes to have those factors. And the way that we were able to test that was PFAM annotation transfer, so domain annotation transfer, and that was presented earlier on, and
So you're
we're able
transferring annotations from these complexity regions.
Yes, we can still transfer annotations to some degree from those annotations. Yeah.
Okay, thank you. So let's thank all the speakers from this